Okay, we're ready to start this procedure. We did a little bit of background about what CODIS is, about what polymorphisms are, the different types of polymorphisms that you can see. It can just be one little bitty base pair, or it can be a short little segment of base pairs, or it could be what we call the variable or the larger assortment of base pairs that we will have changing between person to person to person. And then those 13 loci areas are very important for CODIS, and this is normally what we look for and analyze when we begin running these DNA samples. So in this video, we're going to talk more about the lab procedure parts, and this is going to happen very fast because this is stuff that we've already done for the most part, with the exception of the extraction and the PCR. But as far as making the gel, making the buffer, running samples, comparing the lanes between each other, we've done this before already many times. All right, so in this very first page, this is the extraction of DNA, your DNA, from cheek cells. So what will happen is that you will get some type of tube that's got a saline solution on the inside. And that saline solution, folks, just means salt. That's all that means. So we are going to take salt, and we are going to swish our mouth around, and that salt is going to be spit back down into a cup. We hope that you don't drink or eat anything prior to showing up to this lab experiment. We're going to try to catch you before that goes on, and there's going to be a little bit of waiting period there anyway by the time that we get into the lab and do stuff. So that way we can get as clean as we can sample from your mouth and from your cheeks. So you're going to rinse your mouth for 60 seconds using 10 milliliters of a saline solution, and you spit that back down into the tube that we provide. Next up, it says you're going to swirl the cup. That just helps kind of resuspend the cells and get them swirling around so you can ensure that you get some of them. And it says transfer 1.5 milliliters, probably through the use of a micropipetter, of the solution into a tube with your initials. Next up, it says centrifuge the cell suspension for 2 minutes at full speed. So what will happen is that the solid cells will pack down to the bottom of the tube, and then the liquid just kind of sits up on top. So next up says pour the supernatant, which means the liquid that's above the cells, but do not disturb the cell pellet. So all of that liquid is just really salt solution at this moment, and that salt solution needs to be poured off because we really don't want that for anything. We want the cheek cells that's in the bottom of that centrifuge tube. That's what we're after. Next up, step number 5 says repeat steps 3 and 4 once more. So that means another 1.5 mils goes into a tube, you spin it down, pour off the supernatant, another 1.5 mils goes into the tube, spin it down, pour off the supernatant. That allows us to get enough cheek cells so that way we can do something with it. Next up, 6 says resuspend the cheek cells. So you've got this pellet in the bottom of the tube, and you're going to add 150 microliters of what we call the lysis buffer. That lysis just means to cleave and break open. So the purpose of that buffer is to take your cheek cells and burst them open like little water balloons and get all of that inside stuff out, because that is our DNA plus the other stuff. And it says that that might need a little bit of help, so it is okay by micropipetting up and down. Those cheek cells mixed in with that lysis buffer, go ahead and feel free to use that micropipetter as a mixer or a stirrer or a magnet in a way. Suck it up, squirt it back out. Suck it up, squirt it back out. That is not a big deal at all, and it's pretty common actually practice in a laboratory environment. So in red letters it says note, make sure that the cell pellet is fully resuspended and that no clumps of cells are actually still there. So you do want all of these water balloons to be bursted open as much as possible. So make sure you do a really good job mixing the cells as well as the lysis buffer with each other. So it says put a cap on the tube and incubate it at a 55-degree water bath or dry bath for 5 minutes. So you're going to see that dry bath probably in our laboratory cut on, and at 55 degrees, it's going to be ready for you when you need it. Just put your tube into the heat block and then put the lid on it. Let it do its thing. 5 minutes here, go back to it. Not a big deal at all. Then it says take the top back off, mix it, flick it, vortex it, do what you need to do with it in order to mix that sample up fully. So it says do that for 20 seconds, give or take a little bit. After that, we're going to take that same tube, and this time we're going to put it in a 99-degree water bath for 5 minutes. Again, 99-degree water bath could also be a heat block, so we can take our sample tube, put it into the heat block at 99 degrees, let it sit there for 5 minutes, and this allows those cells to continue to burst open and do what they need to do. Here in red letters, it says make sure that you use a screw cap. We're actually going to start off with this anyway. So the screw cap is there for a reason, and that's to help prevent those PCR tubes from popping. Not PCR tubes, but microcentrifuge tubes from popping their top. And the reason for that is because we want to make sure that the lysis buffer, which is maybe made of water and buffer, right, from boiling off and escaping. We don't want that to happen. So then it says centrifuge for 2 minutes at full speed and transfer 50 microliters of this substance or this liquid to a clean microcentrifuge tube. And then it says place it in an ice bath until you're ready to go to the next section. So here it says the extracted DNA is now ready. That's all that it requires. A little bit of buffer, a little bit of heat. That's it. Some centrifuge, no more. And then we are getting ready to amplify that in the next part of the procedure. So if you're not ready for that, then go ahead and put them in the freezer until you are ready to go back to the DNA sample and pick up from that point. So that's normally where we will stop on the very first day, because going through these extractions and centrifuge steps normally does take up our 3-hour time block. By the time we get in, by the time we realize where everything is, 3 hours are going to go by. So the next lab period is normally the amplification step, and that's what you're going to see here in part 2. So in the second part of the experiment, this is where the PCR amplification is going to come in. So there's a chance that you're going to have to go to the freezer, pull out your DNA sample that you just extracted maybe the week prior, let it thaw to room temperature before this process begins. So this is really where the PCR, which is going to be the newest concept of the electrophoresis labs that you're doing, is going to get initiated. We'll look at the thermocycler. We'll make the settings on the thermocycler the way that it tells us to do here in the lab procedure and the directions. And then there's a lot of waiting that happens after this fact. So number one, it says that you're going to obtain your extracted DNA that you just did in module number 1, the first part of the lab, or the week prior. You're going to take that DNA, you're going to make sure that it's thawed, allow it to come to room temperature as much as possible. And then number 2, it's going to say label a fresh PCR tube with your initials. So that way it doesn't get mixed up when we put it into the thermocycler. Number 3 is going to be the most important piece, because it's going to say add 5 microliters of your extracted DNA. So that is from the week prior. 5 microliters get sucked out and squirted into a PCR tube. And then in addition to that DNA, that's what we want to replicate. We're going to have a few things to allow the replication to take place. First off, we have to make sure that we tell it where we want it to replicate and cleave. Okay, because we're going to have a cut site, and that cut site is going to be determined by our primer. So out of our maybe areas of DNA that we're maybe wanting to know more about, we've picked one to keep this process very clean and very simple, and that is our D1S80 that we've mentioned prior in another video. So 20 microliters of that primer is going to go in to your DNA from your cheek cells, and we're going to mix it together. The purpose of that primer, again, is to recognize the DNA area that has that particular area that we want to analyze on an electrophoresis. So that primer is going to be our starting point, and it is our red or white flag that we are using to say, hey, replication needs to start here. This is where we want to know more information about. So TAC and DNA base pairs, come on in and do your job. So that is the purpose of the primer. Again, there's tons of different primers that are out there. This is just one of them, but the primers normally have to be added separately because there's so many different choices, and there's not really what I would call a universal primer that we just go to and use for these purposes. It really is select what you need for that particular ingredient. Finally, we will also have what we call a PCR EDVO bead or PCR bead. These are typically all the other ingredients that you need. It's very nice. It's very convenient. You know, back in the day, that EDVO bead used to be a series of different ingredients that we would have to mix together. So that would include maybe the magnesium. It would include some of the buffers. It would include the base pairs that we're after. It would include the Taq polymerase. I mean, it would be a whole hodgepodge of different ingredients that we would need to add to these PCR tubes. So over time, we've made this more convenient, and now in a laboratory, we can use what we call a PCR ready bead, EDVO bead, PCR bead itself, and this is all of the ingredients that you need for this replication process to happen. So we will add one bead, we will add your DNA sample, and we will add the primer. And we want to make sure that that bead is fully dissolved, because if it's not fully dissolved, that means the base pairs are not freed up, the Taq polymerase is not freed up, all of my buffer ingredients are not freed up. I mean, that's the purpose of using the bead. So it says mix the PCR sample, make sure the bead is completely dissolved, and to help you with that, the final solution should be a very light orange in color. That is just a visual representation that will help you with this to know that it's fully mixed and dissolved. That's really all that that's for. Number 5 says centrifuge the sample to collect the sample at the bottom of the tube. And then it says amplify the DNA using the PCR guidelines below. So what's going to happen here is that the purpose of the centrifugation step is to pull that DNA down into the bottom of the tube, because that's where the tube is going to be coming in contact with the PCR hot block. So if you are not careful and your sample is like up toward the top of the PCR tube, it is not going to come in contact with the PCR thermo block, and that thermo block is what's controlling the temperatures, allowing the replication process to happen. So you want to make sure everything is pulled down in solution, and you want to make sure it's toward the bottom of the PCR tube, and that's why we centrifuge it here. Because notice it says centrifuge the sample for a few seconds to collect the sample at the bottom of the tube. It doesn't say anything about pouring the decant off. It doesn't say anything about separation here. It just says spin it down. That's really all that they want you to do. And number 6 says amplify the DNA using the PCR below. So here's our thermocycler. Ours looks a little bit different. Ours is not as new as what you're kind of seeing in here. We've had ours for a handful of years. The initial denaturization step is going to happen hot, and that's at 94 degrees for 4 minutes. So that's going to be the very first thing that we do, 4-minute head start. Then it's going to kick in, and our cycles begin to happen. So our cycle is staying at 94 for 30 seconds. Then it goes to 65 degrees for 30 seconds. That's where the primer is going to attach. And then it's going to go to 72 degrees for 30 seconds. And that's where Taq and the base pairs begin to replicate what they need. That is one full cycle. Then it goes back to 94, 65, 72. Back to 94, 65, 72. Back to 94, 65, 72. And it will do that a total of 32 cycles. That's what the directions are telling me to do, and that's what we'll ensure when we get into the lab. Once those 32 cycles are completed, my replication has now come to an end. So my final extension, this is just an extra little push to give time for these things to settle out, come to equilibration, kind of do what they need to do, make sure all the cleanup work is in process. That's at 72 degrees, which is your last temperature, and we hold that again for another 4 minutes. So we've got a 4-minute initialization step. We have a total of 1.5 minutes for every cycle, and we do that for 32 cycles. So that is 1.5 minutes times 32 cycles. So that looks like it's going to be less than an hour. and then the final extension time for another 4 minutes. So on paper, this looks like the PCR replication is going to take an hour, give or take a little bit, right? But the problem is that this heat block has to heat up, cool down, heat up, cool down, heat up, cool down. And that takes a little bit more time. So on paper, your cycle time is going to be a little bit different in total because the thermocycler needs extra time in order to heat up to the temperature and cool down to the temperature. So just be aware of that, and your thermocycler will tell you how long this process is going to take, because it knows traditionally on average how long it takes to heat up and cool down in between each one of these steps. All right. So after that 4-minute cool down phase, we now have replicated DNA that we are ready to go through an electrophoresis on. So that is the newest step that we are doing compared to the other 2 lab experiments that we've done at this time. If we run out of time, it says that you are more than welcome to place your DNA sample back into the freezer at this moment, and that will store it, and it will be ready to go by the time that you come back if a 3rd session is required. All right. In the next session, or the next part of the lab experiment, this is where our gel electrophoresis comes in. I'm not going to spend a lot of time on this section at all. It's pretty much the same stuff. We're going to take our agarose, we're going to mix it with buffer, we're going to make a solution that looks like water from top to bottom. It should be completely dissolved. We cool it down to 60 degrees. We pour it into our casting tray. And then when we pour it into our casting tray, we wait for a few minutes for that to solidify and gel up. Then we will take our samples and we will inject our samples into the electrophoresis, and we allow it to run for a certain period of time. Down here at the very bottom, just like on all the other lab experiments, you're going to see the typical dimensions of the casting tray. We have a 7x7 that we use here. It tells you how much buffer to use. It tells you how much agarose to use. And it also tells you how much diluted cyberstain to use, which is in step number 5. So let's kind of take a look at that, because that slightly is different compared to some of the other electrophoresis steps that we've done. So number 3 says cool the agarose by carefully swirling the flask to promote the dissipation of heat. Number 4 says while the agarose is cooling, seal the ends of the casting tray with rubber end caps. Place the comb in the notch. Then number 5 says before casting the gel, add diluted CyberSafe to the agarose and swirl it to mix it. See table A. So this is a concept that's a little bit different than what we've done before. This cyberstain that we're using in this particular lab is going to be mixed in with the agarose to make the electrophoresis gel. We've not really done that before. So we are mixing the gel electrophoresis agarose with what would be a stain to maybe later down the road save a little bit of time when we go to our staining process. Then it says pour the cooled agarose into the solution, into the tray. Remove the caps and comb, just like you did before. And then place this gel inside of your electrophoresis tank. Diluted buffer goes in, make sure everything gets covered. Sample goes into the wells, just like before, there's no difference. Run it for a certain length of time. So here it tells you the voltage, the minutes, if you have a 10x7, or the minutes if you have a 14x7. Ours is always kind of wishy-washy. We'll just visually take a look at it and see what's going on. After the electrophoresis is complete, remove the gel from the chamber. Keep in mind, when the gel goes in, the wells should be toward the black anode, and the foot of the gel needs to be toward the red, which is the cathode. Negatives are going to be part of the DNA chunk. Those negatives go to the positive. Next up, it says visualizing the cyber gel. Slide the gel off the casting tray, put it on a transilluminator. Number 14 says turn the unit on. Notice it doesn't say anything about staining the gel, and that's because this is embedded into the gel itself. It also doesn't say anything about putting it in water to extract the stain out. This is something that's a little different compared to some of our other two. This process works just as well, which is another reason that we like to do this. The DNA should appear as bright green bands on a dark background, and it says take a picture of your results. Number 15 says turn the unit off, remove, dispose of the gel, cleans the illuminator, blah, 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 blah. So this is our destaining and staining process, folks. You already have kind of done staining while you've been running your gel, and then there's no destaining process. It's part of the gel, and it's attaching to the bands anyway. So this is going to save us a little bit of time on the back end when the staining and the destaining process is completed. All right, so that's the lab directions. I mean, that's kind of what we're after. You'll see a couple of questions that will show up on a data sheet like you've seen before in some of the other lab experiments. It's not a big deal. But there is something that I want to show you that's not going to show up in your document, but it's going to show up kind of in this one. This is kind of an example of what the electrophoresis would look like. So notice that these are not going to be blue, light blue gels with really dark blue bands. This is more of going to be kind of like a greenish, blackish gel, dark gel at least, with these very vibrant color of bands that show up, where we can visualize them a little bit better, a little differently than the other electrophoresis that we've done so far. In the very first lane, we will often put a DNA ladder. We know the purpose of the DNA ladder. We've talked about this purpose before. That's why we're doing it again for this particular time. And then here in each one of these lanes, these are just DNA samples that have came from different people. And again, we don't want to overcomplicate these bands. We want to keep it simple so that way you can do these comparisons a little bit better and a little bit faster. So it says the results photo show an example of possible PCR products from different genotypes. Students as PCR products, well, you're going to show either one band or two bands with lengths between 400 and 785 base pairs. Let's take a look at that for a minute. Over here in the ladder, your document, your booklet, is going to say the ladder should have these bands present, and these are the base pairs of the bands. So the lowest one here is 200 base pairs. I mean, that's what that says right there, 200 base pairs. So the smallest ones travel the furthest. And then this one is the 400 base pair region. This one is the 600 base pair band, and this is the 700 base pair band, and this is the 1100 base pair band. So these directions are going to say this particular cleavage site that we're looking at typically shows up either one or two bands depending on the people, and then those bands will have different base pair fragments maybe, but they will happen between the area of 400, which is this band, to a little over 700, and that's that band there. So this is the region that we're looking for. Notice that this student or this person has two bands that showed up, and those two bands are between the 400 and 785 range. This person has two bands that show up. This person only has one. This person has two bands that show up in that region, and this person has two bands that show up in that region. This band is a little bit above 700, but it can go to 785, and that's okay. So majority of these people are heterozygous, which means they have two different bands that show up in these primer sites that the DNA has been recognized with. Only one person was homozygous, and that's because only one band showed up in their electrophoresis, and that means their parents, their mother and their father, had the same identical area that showed up on both sides to make their own. So even though that these are heterozygous in these other bands, look at these bands, they show up in a different way. So these two bands show up at a different location than these two bands, and these two bands show up at a different location than those two bands. So those are some of the things that we'll be looking for when we start analyzing this DNA gel. We'll take a look at these various DNA samples from every person in the class. We'll determine if they're homozygous or heterozygous for this particular gene that we're looking for. And then on top of that, we're going to be looking at comparison of base pairs. How many base pairs are associated with your particular fragments that showed up on the DNA gel? And just like before, we're going to use a DNA ladder. We'll plot a calibration curve. We'll measure the distances that we're seeing here for these bands, and we're going to be solving for the number of base pairs for each one of these fragment pieces that show up on the gel electrophoresis. Also, it's going to tell us that the PCR product, we just talked about the 400 to 785 base pair region. That's where we're going to start seeing these bands show up, we hope. But 145 base pairs are going to be constant. That's because 23 and 23 on the front end and the back end, these are going to be the primer locations. So that is what the area of the DNA is going to be recognized. That's going to be the same for everybody because that's what we're focused on. That's the primer that we picked. If the primer is going to do the cutting and the splicing, that's where it's going to happen. So the front end and the back end of that particular snippet will be the same number of base pairs for everyone. And then we have 99 base pairs that say flank the variable region that we're looking for. Okay, so when you add all of those up, you're going to see a total of 145 that's going to be constant. So 256 to 640, give or take a little bit, are going to be within the variable region. And that variable region is going to happen right in here. So again, sometimes people's snippet polymorphism is going to be different than someone else's, and that means that different base pairs are going to be present, and some people will have longer base pairs in this region, more of them, and some people won't have as many base pairs in them, and there'll be shorter base pairs in segments. So that's why we see differences in the electrophoresis gel that begins to get analyzed on the chamber and during our analysis when we begin to look at the DNA ladders and what shows up on our end. So 145 are going to be constant. Everyone's going to have the same there, but 256 to 640 is what's going to be variable in this particular region of the DNA from person to person to person. All right. Okay, so that's kind of the, I guess, conclusion of the story. And this is the beginnings of our very first extraction of real DNA samples, in addition to a real PCR procedure that we're going to follow through with in this particular lab experiment. On top of that, at the very end, it is not the traditional stain and destain process that we've seen with our others. This one operates a little differently, and that's great because there's different ways to stain, there's different staining reagents, and this is just another one of those that we get familiar with. So good luck in the lab. I hope that we don't destroy your DNA samples over the course of 2 weeks or 3 weeks possibly, but hopefully, we'll cross our fingers, you'll get something that looks like the picture here in the very last moments of the lab experiment. Okay, so good luck. If you have any questions, let us know. Good luck on the pre and post lab assignment. There's probably going to be a question set that follows through. So hopefully you've taken some notes, you've paid attention, and I think that you'll be ready to answer those questions after you've watched this video. All right, so until then, we'll see you in the lab. Good luck.